Am I audible and visible? Let me know, then we'll start. I think I am visible. Okay, fine. Good. Okay, fine. Hello everyone. Welcome to my class. I'm myself Dr. Deepthi Karya. I'm MD PhD Physiology and I'm your faculty for Physiology. Here in my channel, I'm uploading videos for the first year MBBS nursing, physiotherapy and homeopathy students. If you wish to complete your whole physiology syllabus, please daily watch my video. I'm daily online at 9:30 p.m. Okay, and you can watch me on live classes as well as recorded classes are also available. So stay connected, stay tuned with me to get all my live classes. Another important announcement here, all my soft copies of all my classes, that is also available. Soft copies of all the topics which I am teaching here, that is also available on the app Vesalius. Okay, so you can download the app. The download is free and you can subscribe. In hard copy also, notes available on the Flipkart as well as Amazon. So you can order on the Flipkart as well as Amazon for all the topics which I am teaching here. In hard copy, notes are also available for the same. Fine. Now let us start today's topic. So yesterday we have discussed uh, transport across the cell membrane. Okay, we have started. Hello Sakshi, welcome to my class. Okay, fine. So yesterday we started our transport across the cell membrane. Second topic, first topic was passive transport, second one is active, and today we'll discuss third one, that is special categories of transport. Okay, fine. So here special categories of transport that we will discuss today. So which are the special categories of transport? Let me just a minute. Okay, fine. So here, which are the special, hello everyone, hello Sakshi, hello Raj Mishra, welcome to my class. Welcome you all. Let us start our today's class. Fine. So in special categories, as there are four different categories of transport. One, that is bulk flow. Second is vesicular transport. Third we will discuss, that is filtration, and fourth is osmosis. We will discuss one by one. Okay, starting from bulk flow. What is bulk flow? Bulk, the name itself suggests, flow is in bulk. Okay, here you can see this is passive transport. Bulk flow is passive transport, also known as diffusion. Okay, so here only difference between simple diffusion and bulk flow is here the diffusion that is in bulk. So the transport that is from higher concentration to lower concentration in bulk. Okay, example is respiratory gases. Okay, not a single molecule will be transported, they are transported in bulk. So this is bulk flow. Fine. Now so this is first one. Second variety that we will discuss, that is vesicular transport. Now what is vesicular transport? Here the transport process that occurs for the large molecule. Suppose the molecule is large enough, so it cannot be transported by any ways, not by active transport, not by passive transport. Okay, so what happens, this vesicle, it fuses with the cell membrane and it enters inside the cell, or it exits out. So that is in the form of a vesicle, it forms a vesicle. Okay, another important thing here is vesicular transport also requires energy. Okay, requires energy, and this energy comes, as we all know, that is from ATP, in the form of ATP. Fine. Now different types of vesicular transport. There are three types of vesicular transport. One, that is endocytosis. Second is exocytosis, and third, that is transcytosis. Fine. Endocytosis, exocytosis, and transcytosis. Okay, three types of vesicular transport we will discuss. So starting with this one, endocytosis. You can see here endocytosis means entry of macromolecule. The molecule is large, so it cannot enter by any ways of transport, but here macromolecule, it enters inside the cell through endocytosis. Okay, this endocytosis is again of three types: pinocytosis, phagocytosis, and receptor mediated endocytosis. Fine, I repeat. First of all, vesicular transport is again of three types: endocytosis, exocytosis, and transcytosis. Okay, and endocytosis again has three varieties. Number one, that is pinocytosis. Second one, that is phagocytosis. And third one, that is receptor mediated endocytosis. Fine. So these are the three varieties of endocytosis. Now we will discuss one by one, starting with pinocytosis. Okay, now what is pinocytosis? Pinocytosis, that is known as cell drinking. Okay, drinking and eating. Eating is phagocytosis. So here you can see pinocytosis, that is for the bacteria or other antigen. Okay, this bacteria or antigen is large enough, it cannot pass through the any of the transport, active or passive or any other transport. Okay, another important thing is sometimes some substances, they are also passing through digestive tract and kidney through pinocytosis. Fine, now what is the mechanism? You can see here this diagram shows mechanism of pinocytosis. You can see first of all this molecule is bounded to the outer surface. Okay, before that there is evagination, pit formation here. Okay, there is a formation of pit in the cell membrane. Now the molecule here, it comes in contact. Okay, then you can see the pit is been closed. Okay, and there is formation of a vesicle. Now this vesicle, it contains your substance. Okay, so so this particle along with the vesicle, now it enters inside the cell. Okay, after that what happens, lysosome fuses with the vesicle and it forms phagolysosome and it destroys the particle. Fine, so this is the mechanism of pinocytosis. Fine, next is phagocytosis. Phagocytosis, that is known as cell eating. So here the particle is larger than, particle size is greater than the particle which is pinocytosis. Pinocytosis particle is smaller. Phagocytic particle is larger. You can see solid particle, large particle here. Okay, which enters through phagocytosis. Okay, the particle is larger than macromolecule. Which are the particles that can be phagocytosed? They are bacteria, foreign particle. Hello, good evening, Mahendra. Good evening, welcome to my class. Foreign particle. Okay, and only few cells, they have the capacity of phagocytosis. Like our phagocytic cells are important one, neutrophils. Try to remember neutrophils and macrophages. These particles, these cells, they have capacity to phagocytose the particle. Fine. Now we'll discuss mechanism of phagocytosis. What happens here? First step, that is pseudopodia formation. Pseudopodia means projection from the cell membrane. Phagocytic cell, as we have discussed, neutrophil and macrophages, they first produce pseudopodium. That is, pseudo means false, projection from the cell. Okay, and this is around the bacteria or foreign particle. Now these particles, they are engulfed by this phagocytic cell. This, you can see this kind of vesicle is formed on both the sides of cell membrane fuses, and there is formation of vesicle containing bacteria or foreign particle. Next step is this particle gets fused with the lysosome and there is formation of phagolysosome. Okay, then digestion and degradation of this. Now this lysosome enters its enzymes and destroy these particles. Okay, so digestion and degradation of this content. Okay, and that is excreted out by exocytosis from the other side. So this is the mechanism of phagocytosis. So we have discussed vesicular transport. Okay, this vesicular transport, that is of three types: endocytosis, exocytosis, and transcytosis. Fine. Endocytosis is again of three types. One, that is can anyone? Pinocytosis, cell drinking, phagocytosis, and third is receptor mediated endocytosis. Fine. Now we will discuss third variety, that is receptor mediated endocytosis. Okay, fine. So here in receptor mediated endocytosis, you can see here there are receptors. You can see here particular receptors are present. Fine. Receptor proteins are there, and there is a pit. You can see pit has the receptor, and these pits are having clatherin. So clatherin is a substance, clatherin coated pits are there, which is having receptors here. Okay, now here what happens, how there is receptor mediated endocytosis. This is for particular substance, like suppose some substance, some hormone has to act on the particular cell, so there are receptors for the hormone. The hormone binds with it. Growth factors also transported, toxins and bacteria, they all are also transported by receptor mediated endocytosis. Fine. Now, so how, what is the mechanism? First of all, you can see here this substance binds with the receptor. Or you can say ligand bind with the receptor. So ligand receptor complex is formed. Ligand receptor complex is formed. Fine. So first step. Okay. Then second step is, now this pit, after this receptor binds with the ligand, then there is contraction. Okay. There is a, you can say that clatherin, that is a contractile protein that causes contraction, and this pit, now it gets contracted and it forms a vesicle. You can see here. Fine. Then the whole ligand and receptor along with the pit, now it enters inside. Again, other steps are same. Now your lysosome binds with it. Lysosome causes degradation and release of the ligand and receptor. Okay, here if the substance is hormone, then hormone is not destroyed, but hormone is used by the cell. Hello Kishan, hi, you are late beta. Yes, you are late, but still welcome to my class. Okay, now. So this hormone is used. So this is your receptor mediated endocytosis. Now what is the use of this receptor mediated endocytosis? This is particular for particular substance. Fine. So for example, this is the hormone which is to be bound with this. It will definitely bind with this hormone only, not to anybody else. Okay, Ashish, not to worry. If you are late, better late than never. Huh? If you are late, then also it's fine. Better late than never. But from tomorrow onwards, try to come on time. Fine? Okay. So, okay, good. Now, so this is about, so for those who are coming late, just I'm just telling which we have discussed. We have discussed one, that is bulk flow, or you can watch my class again. Second, that is endocytosis. Okay. Hello, huh? Sorry, Ashish, I'm not getting beta. Issue. Uh, that is regarding app. I think you are asking regarding application. So please, okay, because I'm not managing that. Please raise the question in the Telegram group. Okay, and your issue will be solved. Please raise the question in the Telegram group. There is a Telegram group of the app also, Vesalius app. You can join the group. Link for the this Telegram group, that is also in description box. So first join the Telegram group and then raise your question, because I am not managing that part. Am I clear, beta? Ashish? So, raise your question in the Telegram group of the application. Fine? Now, so, okay, beta. Let us continue. So in endocytosis, again 3 types we discussed: pinocytosis, phagocytosis, and third is receptor mediated endocytosis. Okay, endocytosis means entry. Now exocytosis means exit. Okay, so here you can see reverse of endocytosis. There is a vesicle. Now vesicle fuses with the cell membrane. The membrane is degraded and the substance moves out. This is exocytosis. Okay, very important thing. This exocytosis is present in secretory cells. Some cells, their function is to secrete hormone or some substances. So the secretion is by this mechanism of exocytosis. You can see here, this is the mechanism. First of all, there is formation of vesicle. You can see here. Suppose hormone is there, protein is formed by endoplasmic reticulum. Then this protein that is that is processed, packaged by Golgi apparatus. Then there is formation of vesicle. Now this vesicle comes in contact with the cell membrane, fuses with the cell membrane. Then there is degradation of cell membrane here, and now this protein hormone is released. So this is the way how exocytosis occurs. Now this exocytosis again of 2 types. Number 1 is non-constitutive pathway, or you can say regulated pathway. This one here, what happens, there is in this pathway, this proteins which is secreted, which is you can say processed and packaged in the Golgi apparatus. First it enters, initially enters in the secretory granules, and here processing takes place. Okay, so you can see in Golgi apparatus processing takes place. So when processing is there, that is known as constitutive or regulated pathway. Okay, and non-constitutive or regulated pathway. And constitutive or non-regulated means there is no processing, directly it is released. So that is, these are 2 pathways: constitutive or non-regulated, and regulated or non-constitutive pathway. Okay, if you remember, this is fine. So this is second way of vesicular transport. Third, that is transcytosis. Trans means through and through. You can see here, suppose this is a cell. So endocytosis from one side, exocytosis from other side. Trans, or this is also known as vesicle trafficking. Vesicle enters this way, exit this way. Example, suppose some substances which we are eating from digestive tract, it enters first into the epithelium of digestive tract, then into the interstitial fluid, then into the blood. Okay, so it has to enter this side, exit this side. It will not remain in the epithelium. Okay, or other example, suppose we have some bacteria or virus. So here this is your blood vessel, this is your endothelium. So this bacteria virus enters through this way, exit through this way. So this is transcytosis. Fine. Okay. So this is another variety of vesicular transport. We have discussed 3 varieties of vesicular transport. We have discussed bulk flow also. Fine. So now next one, that is we'll discuss filtration. Filtration is a simple word. Filter. Something when we require to filter. This filtration, that means there is movement of water or fluid. Water or fluid moves, passes from higher, this is very important word, hydrostatic pressure. Hydrostatic means water head pressure. Water head pressure. Fine. From higher hydrostatic pressure to lower hydrostatic pressure. Fine. Suppose you have a filter in your home. What happens? What you require is, you require pressure head. If no pressure, no filtration. So filtration is movement of water or fluid from high hydrostatic pressure to low hydrostatic pressure, and filtration takes place in your blood vessel. Suppose this is blood vessel, fluid is filtering out, or commonly that takes place in the kidney. You must be knowing this is the structure of kidney, है ना? This is the filtration. From glomerulus, the filtrate is formed here. So this is another example of filtration. You can see here, these are the various ways where filtration takes place. One of them, that is kidney. Fine. So this is other variety of transport, filtration. Now, one of the very important variety of transport and very confusing, students, they have so much confusion in this transport, that is osmosis. I will explain very nicely and you can easily understand this. Now, let us start. What is osmosis? First word, first thing to remember about osmosis is a special type of diffusion. Very important thing. So it is diffusion, you have to remember. Fine. So no energy is required. What is the difference between diffusion and osmosis? You can see here. In diffusion, solute and solvent both can pass, whereas in osmosis only solvent can pass, not the solute. So that is osmosis. Fine. Now we will discuss what is osmosis. In osmosis, what happens? Movement of, write down, you can also write down, movement of, very important thing, solvent, only solvent, not solute. Okay, so passive movement of solvent from high solvent to low solvent across the semi permeable membrane. Very important word here, semi permeable. Why semi permeable? Because it is permeable to solvent, not to the solute. Fine. So this is osmosis. Okay, easy way. Suppose I am telling you, suppose this is one solution, this is other, and there is semi permeable membrane here. Okay, now what happens here? Solutes are more, here solutes are less. So what happens? Solvent fluid moves from low solute to high solute, or high solvent to low solvent. This direction, this is osmosis. Am I clear? Am I clear? Fine. Let me know if I'm not clear. I am ready to repeat the things. Let me know, please. If you're watching me live, give me answer in the chat box. You can give me answer also. Am I clear? Fine. Okay, fine. So osmosis, I think nobody is giving answer, means I am clear. So osmosis, that is movement of solvent from higher solvent to low solvent concentration. Now, here sometimes there is endosmosis. Endosmosis means entry of fluid inside the cell. That is known as endosmosis. Okay, fine, Ashish. Good. Thank you. Thanks for replying. And exosmosis means fluid moves outside the cell. That is exocytosis or exosmosis, here it is known as. Okay, now, so for osmosis we have two requirements. Number one is solute concentration must be different. You can see here, higher and lower, one requirement. Second, that is membrane is semi permeable, not permeable to solute, only permeable to solvent. Okay, this is other requirement. Fine. Now, next small definition, that is osmotic pressure. What is osmotic pressure? Osmotic pressure means this, you can see this solute particles, they provide pressure, and this pressure is known as osmotic pressure. How? That is to prevent, why? To prevent osmosis. This is known as osmotic pressure. Pressure provided by solute particles to prevent osmosis. Osmosis means movement of fluid. Clear? And this osmotic pressure depends on number of particles. More the number, more will be the pressure. It will not depend on size of particle, not depend on type, not depend on chemical composition. So if particle is small but numbers are more will be the pressure. Am I clear? So this is osmotic pressure. Now very important other terminology is colloid osmotic pressure. We will discuss first. Then I will discuss other topics. Colloid osmotic pressure. Osmotic pressure provided by colloids. Now which are these colloids? In our blood we have plasma proteins. Okay. These plasma proteins are colloids. Okay. So when plasma proteins are there in our blood, they provide colloid osmotic pressure, means they will not allow the fluid to move out in the interstitial space. Okay. And this pressure normally it is 25 to 30 millimeter of mercury. Am I clear? This is colloid osmotic pressure. Very important thing, when protein content decreases in our body. I think you might have learned kwashiorkor. Kwashiorkor child is edematous. Why? Because protein is less. So what happens when proteins are less? Fluid gets filtered. It will come in the interstitial spaces and there is edema. Am I clear? So this is your colloid osmotic pressure and its importance. Am I clear? Now we'll discuss some other terminologies. That is, we will discuss here. Osmole. What is osmole? Osmole, that is molecular weight of substance. You have to remember this definition. Substance in grams divided by number of freely moving particles. Fine. I will explain. Suppose you have sodium chloride. Okay. So what is the molecular weight of sodium chloride? Sodium and chloride. That is the molecular weight of sodium chloride. It contains 2 moles. So here this is 2 osmole. Molar solution means 1 mole of sodium and 1 mole of chloride. That is there in the molar solution of sodium chloride. Okay. So molecular weight of substance in gram divided by number of freely moving particles. So if there are 9 moles, these are not 9, you can say it, they are 18. If we are talking that we have 9 moles of sodium chloride, what does it mean? Because freely moving particles are double, so 2 osmoles. So your osmoles becomes, if it is 9 mole, osmoles are 18 osmoles. Am I clear? And 1 milliosmoles is equal to 1 by 1000 1 milliosmole is equal to 1 by 1000 osmoles. Now very important other terminology, osmolarity and osmolarity. Easy, I will give you trick to remember. Suppose you have 2L, osmolarity means osmole, number of osmotically active particle or osmoles per kilogram. मीन्स 2 टाइम्स अगर एल आ गया है तो अभी एल नहीं लेना है, और अगर एक ही बार एल आया तो एल लेना है, इज इक्वल टू ऑस्मोल्स पर लीटर. एम आई क्लियर? सो इजी वे टू रिमेंबर, you have to write down 2 times L here. Means, I repeat, what is osmolarity? Osmolarity means number of osmotically active particles जिसे हम क्या कहेंगे? ऑस्मोल ब्रैकेट में. ओके. पर अगर ऑस्मोलालिटी है तो पर किलोग्राम आएगा, क्योंकि एल 2 बार आ गया, अभी एल नहीं लेना है. इजी वे टू रिमेंबर. एम आई क्लियर? ओके. एंड ऑस्मोलारिटी मीन्स ना ऑस्मोल्स पर लीटर. एम आई क्लियर? फाइन. Now another terminology is isotonic, hypotonic, hypertonic solution. Now what are these solutions? Isotonic means osmolarity equal to plasma. Okay, normal osmolarity of plasma, that is about plasma की osmolarity कितनी रहेगी 270 to 290 milliosmoles per liter. If you are taking some solution which has osmolarity equal to plasma, that is known as isotonic. Okay, suppose if we are placing RBC in the isotonic solution, there is no osmosis, so RBCs are normal. Fine. Second, hypotonic means when osmolarity is less than plasma. Suppose RBCs are placed in hypotonic solution, what happens? Osmolarity less here, more here. So fluid moves. Okay, Ali. Okay, you are late here. Fine. So osmolarity is less means fluid moves from outside to inside. So RBCs they swell. So there is swelling of RBC if we are placed in hypotonic solution. Hello Ali, welcome to my class. Watch the class later on if you are late today. Okay, save the link and watch it later on. Fine. Okay, then hypertonic fluid. Here when RBCs are placed in hypertonic means osmolarity more than plasma. And when we are placing RBCs in a hypertonic solution, what happens? Hypertonic means osmolarity is here more, here less. So fluid moves from RBC to outside. Always remember, fluid moves from hypotonic to hypertonic means less solute to more solute. Am I clear? So this is hypertonic fluid. Okay. Now, okay, thank you, beta. Now next is applied aspect here. Suppose somebody has severe dehydration. What happens to osmolarity? Because fluid is lost. Fluid is lost. So what happens when fluid is lost? Then osmolarity increases. Okay. Suppose in diabetes mellitus, what happens? Glucose, blood glucose level increases. Again, osmolarity increases. And because of increase in the osmolarity, what happens? There is coma. Why? Because of fluid flows from inside the cell to outside. So in brain cells, if fluid comes out, that results in decreased function, hyperosmotic coma. Okay. Then when urea level increases in the patient with renal failure, again same thing, hyperosmolarity. Okay. Then another is hypertonic. Sometimes the person, this is clinical aspect of today's class. Sometimes in case of edema, if some patient is having cerebral edema, edema of the brain tissue, as a treatment, mannitol drug is given. Now, what is the action of mannitol? Mannitol remains in the blood vessel. It will not come out of the blood vessel and it moves the fluid inside the blood. So, what happens? Fluid from the interstitial space, now osmosis of the fluid takes place. So, this interstitial fluid decreases and edema decreases. So, this is the mechanism of action of the drug mannitol, which is given in the patients of edema, cerebral edema. Fine. Last topic, that is solvent drag. What is solvent drag? Sometimes what happens, along with solute, your water also moves. You can see not only solutes are moving, water is also moving, and that is known as solvent drag. Okay, some other applied is sodium potassium pump abnormality. When sodium potassium pump is abnormal, that results in certain disease condition. Like one, sodium potassium pump abnormality disease results in heart failure, results in hypertension because of the excessive reabsorption of sodium from renal tubules. Fine. So this is about the applied aspect. Okay. Now we have shortage of time, but still I'm trying to complete 10 MCQs of today's class. Okay. So be quick, answer the MCQ. Okay. Let us start. I'll give 30 seconds only for each MCQ. Fine. First MCQ time starts now. The process by which macromolecule enter due to fusion of part of a cell membrane enters. Okay. Time starts. You can give answer in the chat box. Can anyone give me answer? B. Very good. Ashish, good answer. It is endocytosis. Very good. Next, uh, okay. Next question. Exocytosis means extrusion of cell-bound vesicle, intrusion of lipid molecule, intrusion of solid particles, all of the above. Exocytosis, beta. Kishan, exo, not endo. You are giving, I think, first answer. Fine. Sorry, give me the second answer. Just a minute. Just a minute. Let me see. A, very good. Ashish, good answer. Extrusion of cell-bound vesicles. Good. Third question. Which of the following is used for exocytosis? Potassium, sodium, calcium, phosphate. This I think we could not have discussed. But the answer also you can guess mostly. Uh, which of the following is used for exocytosis? I'll give you the answer because we could not discuss. And the correct answer is calcium ions. Calcium. Now, next. Clatherin that is used in endocytosis, exocytosis, receptor mediated endocytosis, and transcytosis. If you're live, please give me the answer. No, okay, I'll give the answer. Clatherin is used in this one, receptor mediated endocytosis. Fine. Next. Next question. Endocytosis. It is the process by which extracellular substances are engulfed. It includes pinocytosis, vesicular transport and does not require energy. Process by which intracellular substance is removed out of the cell. If you are watching me live, try to give answer. If you cannot, I will give the answer. Not to worry. Here the answer is first one, this one. Process by which extracellular substances are engulfed by the cell. Fine. Next. Transcytosis occurs in epithelial cells of intestine, neurons, neuroglia, and exolemma. This is easy answer. You can be able to answer me. I think so. What is transcytosis? No, okay, I will answer. This is present in the epithelial cells of intestine. As I told you, suppose some substances are transported from intestine to the interstitial fluid, then the blood vessel. So, one side there is endocytosis, other side there is exocytosis. Fine. Next. Which of the following is not vesicular transport? Uh, Sorry, I couldn't get all your responses because of internet issue. Fine. Okay. Six number A, that is correct, beta. Ashish, you are correct. Fine. Seventh. Now give me the answer of seventh one. Question 2A, that is also correct. Fine. Now write down number of question and answer. So that will be easy for me to get it. Very good. 7D. Very good. Kishan and Ashish. Correct answer. Good. Number 8. Very good. I'm getting good responses. I'm happy with your responses. Okay. Number 8. In which pathway proteins are transported to the cell membrane in the vesicle with little or no processing? Okay. No processing. That is A or B? A or B? Which one? Give me answer. Kishan or Ashish, if you're there. No. Yes or no? If you can be able to answer, that is fine. B. Very good. Very good. It is correct option B. B, beta. B is the correct option. I will give you hint. I will give you trick to remember. No processing. एक बार n आ गया ना, अभी n नहीं डालना है. So constitutive. Okay. So n must come once only. Processing, then non-constitutive. Am I clear? So you can remember now. No processing. No processing, so the answer is constitutive. Processing, then non-constitutive pathway. Am I clear? So this way you can easily remember. Ninth question. Which changes occur in the RBC when placed in the hypotonic solution? Here, RBC and hypotonic. Here solutes are more. So what happens? Swelling or shrinking? Tell me. Hypotonic. Fluid moves from hypo to hyper or hyper to hypo? Tell me. Fluid moves from... No, it is not correct, beta. Kishan, it is wrong. That is RBC is this swell. Swell. A, B is the option, not A. Here fluid moves from hypotonic osmosis inside. So RBC is this swell. Okay. Here you can see. No, beta. Option is B. B is the option. Hypotonic. Always fluid moves. Try to remember, fluid moves from hypo to hyper. Hypotonic means fluid is more, solute is less. Barabar? Fluid more and the movement is this way. Fine. Okay. Now last question. Osmolarity. I told you osmolarity and lity. Riti is equal to osmol per liter or per kilogram. Tell me. I told you L comes 2 times always. This is the trick to remember. Here osmola વધુ થી ઓછા તરફ. Okay. From higher to lower, solvent. Higher to lower, solvent. Kishan. That is very good. This is osmoles per liter. Very good. Ashish, correct option. I told you. Osmolarity. Okay. Riti means here 1L. So you have to find out osmol per liter. So this is 2L. Now if the word is osmolarity, okay, so this is per liter. And if the word is osmolality is equal to, hello Gilles, welcome to my class. Osmolality means osmoles per kilogram. You are late, Gilles. I think we are towards the end of the class. Okay, this is our last MCQ. So please come on time from tomorrow onwards. Fine. Hope you are coming early and watch my class, beta. Okay, fine. So this is about our today's class. Thank you. Thanks for watching me. Again, announcement that all my topics notes, they're available in soft copy as well as hard copy. To get the soft copy, you can download the app Vesalius and you can subscribe. And for getting hard copy, already that is available in book, that is Physiology notes, that is available on Flipkart as well as on the Amazon. Okay, so stay connected. I'll be taking classes daily from Monday to Saturday, and we are going to complete all the topics. Okay, thank you. All the topics of physiology. Thank you, Kishan. Okay, thank you so much. If you like my video, you can like it. You can share with all your friends, those who are ready to watch and learn physiology. Okay, and subscribe my channel. Thank you so much. Thank you, Avni.